Hi guys, thanks for joining on time, but we'll just wait for like 2-3 more minutes for anyone else to join, then we'll start the class. Yeah. Hi, good evening. We're just waiting for another 1 or 2 minutes before we start the class. Okay, I think we can start today's class and people will join as we go. So I'll just share my screen with you guys. Yeah, I hope that is visible to everyone. So I think we can get started. Yeah. All right. So, welcome everyone to this week's problem solving session. Oh, sorry, this should be week 6. I just realized. Yeah. So, which is gonna tackle concepts from the respiratory system that were covered in the coursework. So last week, I'm not going to spend too much time recapping because there's quite a lot to cover this week. Last week we just talked about the excretory system and the different important mechanisms involved in um, making sure that the correct amount of fluids and salts are reabsorbed or excreted via the kidneys. So we talked about different reabsorption mechanisms that are distributed the functionally between the different parts of the nephron, from the glomerulus into the Bowman's capsule, the PCT, loop of Henle, the ascending and descending loops, limbs of the loop, and the DCT and the collecting tubule. And we learned about the different types of ion channels that are present there and the permeability to salts versus water in these different parts. Um, which contribute, and obviously how there's a hormonal control that allows the final product, which is the urine, to have the correct osmolarity, the correct amount of excreted products in it that make sure that the blood pressure and the blood osmolarity is maintained. Meanwhile, excess things are removed from the body. So if you want to check that out, it's already posted. So today we'll directly jump into talking about the respiratory system, which is the new system that we're studying now. So as you all know, the respiratory system is just like every other system, it's highly, highly essential because it is primarily responsible for providing oxygen to cells and tissues in the body, right? And removing the carbon dioxide generated during respiration in all of the cells. So within the respiratory system, this gas movement from, as you know, it moves from the atmosphere into our lungs. It moves through these different stages, which I'll discuss now, and eventually reaches the alveoli. So we have the upper and lower respiratory tracts that facilitate the movement of air from outside to inside. So we start by taking in air from the nose, the nasal cavity, or the mouth, right? And this moves, and even it moves into like the sinuses as well, and it moves through the throat and via the windpipe. And we have the larynx on the way that, of course, as you know, allows us to make sounds because of the vibrating vocal cords. And Then the air moves from the trachea, which is the windpipe, it moves into the lower respiratory tract. And that's made of the lungs, which you can see here, and the large airways, which are the bronchi, which are essentially the larger tubes. And then those branch into bronchioles, which are tubes with smaller diameter. And finally, the bronchioles branch and branch and branch further into these little sacs called alveoli, right? And these alveoli have very, very thin walls that allow for gaseous exchange with all of these blood vessels that are surrounding them very intricately, right? So this gas movement that's occurring when the air moves into the alveoli and then needs to diffuse into the blood so that the oxygen and carbon dioxide can be exchanged through the mechanism of hemoglobin based transport, right? That is all based on partial pressure, meaning that gases will move from high partial pressure to low partial pressure, ultimately moving to the mitochondria in the cells. When the hemoglobin carrying this oxygen diffuses into tissues and is releasing this oxygen, ultimately the point is that it moves via a gradient of partial pressures from outside to all the way to the mitochondria of different cells in the tissues via a gradient of partial pressures. Okay, so this overall organization over here is something I thought I could outline for you guys. So I think that's fine. So the number of alveoli, actually, I believe there's like more than, I think, 200 to 300 million alveoli. So the total surface area that alveoli would cover is like huge. So the amount of surface area that's available for gaseous exchange is gigantic, right? It's like the size of like a tennis court, I think is what was mentioned. And all of these organs and all are housed inside the thoracic cavity and protected by the skeletal structure called the rib cage. And the rib cage are joined, like the muscles that help with the movement of the rib cage, which allows the lungs to expand and contract during respiration or during ventilation, are the intercostal muscles. You have external and internal intercostal muscles that are between the ribs. Yeah, so that's that. Then, if you look at the respiratory system with respect to at a more cellular level, like how the tissue architecture is there, you will know, okay, so the lungs are within the thoracic cavity as seen, and then they're connected to the trachea and the bronchi, and they are surrounded by this tissue, which is called the pleura. So you have the visceral and parietal. So the visceral is more inner, and the parietal is the outer layer, and both of them are called pleural membranes or pleura. With sorry, with airflow driven by the diaphragm movement. So we'll talk about that in a minute. So the bronchi, right, which are the largest tubes in the lower respiratory tract, they are lined by this type of cell, this type of tissue comprising of ciliated columnar epithelial cells. And they also have these special cells called mucus secreting goblet cells that are very important because they lubricate the passage and trap particles. And the cilia obviously are very important for sweeping away dust and debris that might enter, and mucus traps them. And you also have cartilage and smooth muscles which maintain airway structure and regulate airflow. So it's very important that this cartilage, especially in the larger airways, is present because it prevents airway collapse. Alveoli, interestingly, they don't have any cartilage because they need to just remain open with the thinnest layer such that proper exchange can take place. But in these, because there is constant ventilation, it's very important that there be this cartilage that supports it. Over here, you can see these rings of cartilage that support the structure to prevent it from collapsing because there's going to be negative and positive pressure as we breathe, and that shouldn't cause airway collapse. Right. And you have different types of cells. The ciliated cells, you have goblet cells, you also have some basal cells, which are like stem cells that can replace any damaged epithelium. You can have some club cells that help to clean up, neuroendocrine cells that help sense carbon dioxide partial pressure, so on and so forth. And yeah, so then when you transition to the respiratory bronchioles and alveolar ducts, the conducting airways become gas exchange regions. So you're becoming, essentially, it's becoming like thinner membranes or thinner layers of cells that allow for this exchange. And finally, you reach the alveoli, or singular is alveolus, where you have these type 1 and type 2 cells. So the thin type 1 cells are the ones that help with gas diffusion, whereas the type 2 cells are important for secreting surfactant. And surfactant is a type of like material that prevents collapse, and it surround and essentially helps maintain the structure. And then surrounding these alveoli, you have capillaries carrying blood, and that allows the oxygen carbon dioxide exchange between air and blood at the level of the alveoli. So that's about the sort of cellular structural organization of the respiratory system, right? Um, yeah, so I think that's sort of the summary I had. The alveoli themselves, they have like a diameter, they're really, really small, okay? Like you can have one big cell that's maybe like half the size of this alveolus. So I think they're like 100 to 200 microns in diameter. So like literally so small. And it's a very dense capillary network for gas exchange. Um, yeah, I think that, oh yeah, one thing I remember that I should mention, which is that these type 1 cells, these are like the majority of the cells. So they're like 90% of the alveolar cells are actually these type 1 pneumocytes. 90% of them, right? And they are the ones that with the thin squamous epithelium, mainly for gaseous exchange, whereas the type 2, they are the remaining 10%, and they secrete these surfactants, essentially some types of phospholipids, and they reduce the like surface tension, which is what surfactants do. They reduce the surface tension and prevent the collapse of these alveoli. Yeah. So that's pretty much it. I think we can move on to some questions, and then maybe we can discuss as we go. Right. So the primary driving force for oxygen movement from alveoli into blood is which of these? So for anyone who still like has not attended before, you can just either say your answer out loud, or you can send it in the chat box and we'll discuss it. So please go ahead and try this question. And yeah, I hope everyone can hear me. Does anyone want to try?
Diffusion along partial pressure gradient.
Uh-huh. Does anyone else want to give it a shot? You can send also if you're not comfortable with speaking out loud. Let me see. Okay, nice. Yeah. So you guys are absolutely right. The correct answer is diffusion along a partial pressure gradient. So, um, as you know, what is a partial pressure, right? So essentially, it is the pressure exerted by, say, if you have a mixture of gases. So, like in the air as well as in our tissues, we don't just have one gas. We don't have only oxygen or only something, right? You have many different gases: nitrogen, oxygen, carbon dioxide, right? So the partial pressure of a gas is essentially a measure which tells about the pressure exerted by one of the gases in this mixture of gases. So, in the air, we have 21% oxygen and 78% nitrogen and some, whatever, 5% or whatever of carbon dioxide. Um, so every gas here contributes its own share of the total pressure. So you have this law called Dalton's law that talks about the total pressure, which essentially adds up all the partial pressures of the mixture of gases, and that gives you the total pressure of that mixture. That's Dalton's law. So at mean sea level, you have a total atmospheric pressure, which I think you guys know it's about 760 mmHg. So that is the composite of all of the partial pressures of the different components of the atmosphere. So different gaseous components of the atmosphere, right? So oxygen's partial pressure would be, 21% of that total amount. Carbon dioxide would be, whatever. So each one you can calculate the individual partial pressure of air. Obviously, this would be subject to many things: temperature, altitude, humidity is a major factor, right? So why is this important inside the body as well? Because in the body as well, in tissues, you have partial pressures and gases will move according to those partial pressures and not like the total pressure of all the gases. So oxygen will always diffuse from, say, high partial pressure to low partial pressure until it reaches an equilibrium. That's the only way it's able to move from the outside, or basically the air in the alveoli, to into the capillaries, into then the tissues, and then into the mitochondria. So in the atmosphere, you have about, for oxygen, your partial pressure is about 160. In your alveoli, it's a little lower, so 100. So this gradient allows oxygen to move from the atmosphere to the alveoli, and then it moves from the alveoli to the arterial blood, which is a little lower, and then the arterial blood to the tissues, which is even lower. So I think the tissues are like, I think maybe like 30 or 40 mmHg. I'm not exactly sure. And then from the tissues moving to the mitochondria, of course, that is like a very big gradient, because mitochondria will be so low partial pressure of oxygen, like 1 or 2 mmHg. So this diffusion gradient is maintained at every single step of oxygen movement, and this is due to the differential partial pressures, and that's what keeps this oxygen moving from when you breathe in to when the oxygen reaches your tissues. Okay, so yeah, this is about, yeah, so there's not really any active transport like we had studied about how ions and so on. There's like active transport, like for this gas, there's no pumping or active transport. It's just diffusion. It's passive, and it's because of this partial pressure gradient. Okay. All right. So here is a question again. So approximately how many generations of branching occur in the human respiratory tree before reaching alveoli? So here are your options. Let me know what you guys think. Please, everyone, try answering the questions. You can send it or you can say it. This was there in the coursework. It was just mentioned. I thought I would generate a question about it. Okay. Yeah. So good. I see you trying. Others also, please do give it a shot if you know. You can, even if you're wrong, there's like literally no problem. So you're right. The answer, I think you meant B, but like the answer is 23. Yeah. So there's 23 generations. It's option B, okay? 23 generations of branching before um, you reach the alveoli. So there's repeatedly the airways are being branched. So you start with the trachea, right? Then the bronchi, the bronchioles, and then you have many branches, ultimately reaching terminal bronchioles leading into the alveoli. So in humans, there are about 23 airway generations that ultimately lead to millions of alveoli and a huge surface area. Okay. So this is just kind of like a factoid. There's not really any major explanation behind it. I'm sure other animals, though I don't know this, maybe you guys can look it up if you're interested. Like, how much branching is there in other animals? Would a lion have the same respiratory tree branching, whatever numbers as a human would? Like, maybe I can find that out while you guys answer the next question. Okay, so here's the question. Which cells primarily secrete mucus in the respiratory epithelium? So give it your best shot. So, the correct answer here uh, is C option, okay? It's goblet cells. So, the goblet cells in so goblet cells are in general known for secreting mucus, like not only in the respiratory system but in other systems as well. They are the ones that do it. They secrete mucus in the form like mucins, like they have a lot of glycoproteins and stuff in in mucus. It makes them really slimy, right? And they trap dust and microbes and like pollutants of different kinds that might enter. And they protect the epithelium from drying up because you need to keep it sort of moist and protected. Basal cells are actually like the type of stem cells, I believe, that help with repair of the epithelium of the respiratory system. And type 1 cells, as you know, are important for a gaseous exchange at the level of the alveolar. And Clara cells, what did the Clara cells do? Forgetting. Hmm. I think the Clara cells are important for cleaning up. But let me just check that. I'll get back to you. But the answer here is option C, which is goblet cells. So I hope that's clear now. If anyone's confused about anything, just let me know. So we'll move to the next question. So the main function of cilia in the respiratory tract is to do which of these functions? So let me know what you guys think.
Option D, ma'am.
Uh, yeah, you are absolutely right. I think all of you got it completely correct. Yeah, good job. So the cilia are very important to move the mucus along and any trapped particles towards the pharynx so that they can be thrown out because they shouldn't go further down into the lower respiratory tract. That could be really damaging and it could clog up those very, narrow branches of the respiratory system. So they move the mucus along in a kind of like wave-like motion and they sort of push it along with any trapped pollutants and stuff. Uh, so I was just reading about the Clara cells because unfortunately I'd forgotten what they do. So these cells are mainly there in your terminal bronchioles. So I'll just go back to that question, right? So these Clara cells, right? So since we were talking about the different types of cells, so they are in the lower respiratory system terminal bronchioles, and they're actually called club cells now apparently, and they are they're not ciliated and they secrete a protein like a that's special to them, like a secretory protein specific to the club cell that are important because they're anti-inflammatory and they also have some surfactant components and they help with maintaining and protecting the epithelium and reducing inflammation since they secrete anti-inflammatory proteins. So they essentially help to clean up the okay. They have this like kind of detoxification function where they contain a bunch of enzymes that help to metabolize toxins and stuff. So if, for instance, someone is breathing in a lot of pollution, or if someone smokes, God forbid, then this can help to detoxify to some degree. But obviously, any excessive amount of inhalation of pollution or smoke would just override any of these safety mechanisms. Yeah, so they have a bunch of different functions. So these are one of them. Yeah, cool. So we did this question. So what about type 2 alveolar cells? What are they mainly responsible for? So we discussed it. Let me know if you guys remember.
Surfactant secretion.
Mm-hmm. Does anyone else want to try? Yeah. Yeah, awesome. So that's absolutely right. You guys all got it right, which is surfactant secretion. We talked about the two types of alveolar cells, type 1 and type 2. So type 1 is important for gaseous exchange and it makes about 90% of the alveolar cells, whereas type 2, which is the lower amount, that's about 10% of the cells, they are important for, as you said, surfactant secretion. So option C is the correct answer here. And surfactant is sort of like a detergent, okay? So it just prevents alveolar collapse. It's really important. Okay, let's go to the next one. So how does surfactant in the alveoli actually work? So this is relating to that. Give it a think and see if you can figure out what it is. It just sort of depends on what is a surfactant. Like what does it actually do? So if you guys have heard about this word surfactant,
Option B,
uh-huh,
ma'am.
which one?
Option B, I think.
Yeah, yeah.
Reducing surface tension.
Yes, yes, you're right. It is about reducing surface tension. So why is it important, right? So I can just explain that in brief so that it's sort of easier to remember this. So it's as a sort of concept, um, like it's basically physics, right? So why do we need surfactant at the level of the alveolus? Because essentially what is surface tension, right? Surface tension is um, a force that essentially tries to pull the alveolus inwards. Okay. So if you guys remember studying like Laplace's law in physics, where P is 2T by R, essentially P is the surface tension or the collapsing pressure. The sorry, P is the collapsing pressure and T is your surface tension. R which will be the radius of the alveolus. So essentially, if you have a very small alveolus, your denominator is becoming really small. So there's a higher collapsing pressure. Okay, so P and R are inversely proportional, meaning the collapsing pressure will increase the smaller the alveoli are. And the alveolus is really tiny, right? We discussed its diameter. So without the surfactant, something so small, it was just highly likely that it would collapse. Um, and then breathing obviously would be really hard to do if you have a collapsed alveoli. It wouldn't expand properly, and the gas wouldn't enter it properly. So surface tension is something that's constantly trying to pull it inwards, and this is because there's actually like these membranes are not dry, right? So the inner surface of your alveolus is actually lined with a layer of water, and water molecules are what are attracted to each other, and that's what's creating this inward pull of surface tension. And so it's trying to pull the alveolus inwards because of these water molecules that are keeping the membrane moist, but at the same time causing a high probability that it will collapse inwards, and based on Laplace's law. So without surfactant, it could collapse. So that's why these type 2 alveolar cells are secreting this surfactant, which is essentially an amphipathic molecule. So you guys have heard about detergents, right? Detergents are amphipathic, sorry, and they essentially have a hydrophilic head, right? So it's like if you can imagine this is a head and then there's like a tail like this. So the head is hydrophilic, means it likes water, and the tail, it's just a long sort of fatty acid tail that is hydrophobic, which means it repels water. It doesn't like water. And this compound essentially just spreads over the water in the alveoli, that layer of water. It spreads over it like a monolayer, and it just sort of inserts itself between the water molecules and prevents all of this hydrogen bonding, and it reduces the amount of water to water attraction, and that lowers the surface tension. So if you lower the surface tension, you essentially are reducing how much the water is attracted to other water molecules and preventing the likelihood of alveoli collapsing. Okay, so that's why it's really important for these cells to properly secrete um, surfactant and prevent collapse of the alveoli. So that's sort of the physics behind it. And what do these surfactants primarily consist of? So you know how in our membranes, right, our cell membranes, we have phospholipids. So these phospholipids are also amphipathic. So about, I think, like 80-90% of the surfactants are actually just phospholipids. DPPC is one of them. Uh, and it's a very commonly used phospholipid for making like synthetic bilayers. I make bilayers in the lab all the time using a slightly different version of this phospholipid, and they form these like bilayer like materials. And yeah, anyway, so you have these different types of surfactant proteins. Not just that, you do have other things as well, though I'm not too well versed with that. And some small amounts of, I think, cholesterol and things like that. Yeah. So cool. Great. Oh, yeah. And I remember reading about this thing called NRDS, which is like neonatal respiratory distress syndrome. And that's in these preemie babies, right? Babies that are born earlier than they were, supposed to. So they have some problems and they lack surfactant production to an adequate level. So actually their alveoli collapse. And so they to treat them, to save them, the doctors need to give them like some exogenous surfactants or steroids that help to stimulate the type 2 cells to then produce enough amount of um, alveolar surfactant to prevent this collapse. Yeah. So it's just some factoid for you. Okay. All right. We have another question. This is about the sort of neuroendocrine functions within the respiratory system. So you have the activation of these beta 2 adrenergic receptors in the airway smooth muscles. So what would this cause? So I'll explain this once you guys have tried the question.
Option B, ma'am.
Yeah. Would others like to give it a shot? Hmm. Yeah. Great. So the correct answer here is option C. So sympathetic stimulation, like the one which we've described here, results, which is via these beta 2 receptors, can lead to smooth muscle relaxation. Okay, and if the smooth muscles are relaxing, you can have bronchodilation. So essentially, um, people who have asthma, right? So they have to take these inhalers and they take like a little puff from the inhaler and that transmits this like in a gaseous form, like it's like a spray essentially, and it um disputes disperses this chemical. Like I think one of them is called salbutamol, which is there in the asthma inhalers, and that uh stimulates this bronchodilation essentially. So if someone is having like an asthma attack, it means that there's some constriction in their airway, which is causing a problem with breathing, taking in enough air. So they're really like, they're expressing a lot of respiratory distress. Um, and so in those cases, the person can take like a bit of salbutamol through their inhaler, and that triggers bronchodilation. And this is via this sympathetic stimulation or the beta 2 receptors. So that leads to smooth muscle relaxation. So the answer here is option C. And meanwhile, parasympathetic activity can lead to bronchoconstriction, the other way around. Okay. So, yeah, I hope that is clear. We'll move to the next question. Okay, so during inspiration, taking in air, which is the following changes occurs? So this is about the process of the actual physical process of breathing. So if you guys think, then we'll also discuss actually how it happens in the body. Does anyone else want to try? Just think about the mechanics of breathing. Yeah, so good job. Yeah, the correct answer is indeed option D, which is the diaphragm flattens to increase the thoracic volume. So how does this work? So when during the process of inspiration, so you're taking in air, the diaphragm contracts. So essentially, it flattens. When it flattens, you're essentially increasing the thoracic volume. And when you're doing that, the pressure within the cavity is decreasing. So you guys might have studied Boyle's law, right? I'll actually be explaining that in some more detail, so I won't go into it into much. But essentially, you have like an inverse relationship between pressure and volume, according to Boyle's law. So when you're increasing your thoracic volume by flattening the diaphragm, you have a decrease in pressure in the thorax, in the thoracic cavity, and that allows the air to enter the lungs based on a again a pressure gradient, right? So again, as I mentioned, air always moves via these pressure gradients. So this particular pressure gradient sucks the air into the lungs. So the correct answer here is that, yeah, the diaphragm flattens to increase the thoracic volume, right? The diaphragm is at the lower sort of extent of the thoracic cavity, and it is right above the abdomen. It's a very, very sort of strong layer that facilitates constant breathing throughout your life. Yeah, cool. So hope that's clear. So now let's talk about a bit more about this diaphragm. So it receives motor innervation. So primarily, which nerve supplies it with innervation? Yeah. Does anyone else want to try? Okay, so I'll tell the answer now. Uh, oh, so the answer is option C, which is the phrenic nerve. Okay, so the phrenic nerve, it's arises in the neck, right? And it has the C3 to C5. So C3, C4, C5 are these nerve like branches, fibers that are that compose the phrenic nerve, and that's the part that innervates the diaphragm. So there's this there's this mnemonic that I found online, which was C345, keep the diaphragm alive. So you can sort of remember it in that way. So the phrenic nerve originates from the cervical spinal roots, C3, C4, and C5. Okay, and that's really important for innervating the diaphragm. This is just sort of a knowledge thing, but that mnemonic might help you to remember this in the future. Okay, all right. So, all right, yeah, yeah. Okay, so these we did a few questions. Now let's talk a little bit about some interesting concepts. So before we move on, this is my weekly reminder to not smoke because it really, really screws up your lungs. So this is some information that I got here, which is um, that the huge difference between healthy and smoker lungs. So just you can appreciate how different it is. And I've actually seen this because I've like witnessed like an autopsy once as part of a course that I had taken a long time ago. And the the person whose autopsy was being done, that lungs were actually like black, literally. Like they're not even kidding when they tell that the lungs turn black. Like it's actual, it's real. Like they have these deposits of black all across the lungs. It's really gross. So, healthy ones, they maintain the elasticity, they're pink because there's like good blood flow and everything's going good. But with smoker lungs or even lungs that are exposed to high pollution, even they become really dark, rigid, they get scarred. And there's a lot of proper oxygen transfer, but here the alveoli start to get damaged with time because of a buildup of all of these different materials in them and so on. And that can lead to a huge proportion of the alveoli being damaged. So you don't have enough oxygen efficiency. So a lot of smokers actually start wheezing because they don't have like proper amount of oxygen entering, and the vital capacity decreases. So whatever the proper volume is that could be filled inside the lungs, that decreases. There's a lot of capacity loss. And obviously, there's a lot of cilia paralysis, so there's higher infection risk. A lot of smokers develop a lot of infections. Um, so here, whereas in the healthy lungs, there's active clearing mechanisms because cilia are healthy, they're swaying nicely and sort of sweeping the mucus along with whatever pollutants. Here, the epithelia is damaged, the cilia are damaged, so there's a higher chance that pollutants remain there, some microbe remains there, and that can lead to higher infection. And particularly in developing countries like ours, there is a higher chance of getting some respiratory disease because we already have so many microbes in the air and so many people have so many sicknesses, and so to increase the chance of falling ill because of some opportunistic infection that wouldn't otherwise make you sick, smoking really would increase that possibility a lot. And obviously damage recovery is also lower because I'm sure your stem cell layers are decreasing. So smoke from cigarettes can contain tar and toxins, can cause chronic inflammation, emphysema, which you would have heard of, and even COPDs, like chronic obstructive pulmonary disorders. These have deterioration of the alveoli walls, elasticity of the lungs. It makes it hard to breathe, and smokers, they have a large percentage of them get emphysema. And even e-cigarettes, which people think are like better or like okay, in comparison to like the regular cigarettes, like they're also bad. They have chemicals in them that can cause these different types of inflammatory conditions. And obviously, it risks the risk of cancer increases by a huge, huge proportion. So 85% of lung cancer cases is caused by smoking because the carcinogens in um, from the cigarettes, they damage the DNA of lung cells, and any one problem could result in unregulated cell growth. So, yeah, this is just a weekly reminder to not do it, don't do it, and spread the word. Um, yeah, so coming back to the course content, though that's just as important, we'll talk a little bit about Boyle's law. So I mentioned how the partial pressures and the gradients of air or gradients of pressure uh, allow movement of gas from outside to inside. And a lot of this is regulated by Boyle's law in the thoracic cavity, particularly. So we'll start from up here. So the space between the outer surface of the lungs and the inner thoracic wall is known as the pleural space, okay? And this is usually filled with this fluid known as pleural fluid. So it forms a seal which holds the lungs against the thoracic wall with the force of surface tension. So if you see, like for instance, if you have two surfaces that are covered in some water and you put them together, they sort of tend to stick together, right? That's essentially water molecules attracting each other and causing those things to stick to one another. And so this seal that forms actually ensures that when the thoracic cavity is expanding or reducing, the lungs also undergo expansion or reduction in size proportionately or according to that. So while there's breathing going on, this contraction and relaxation of muscles acts to change the volume. So that's about the pleural space, right? So this is about the thoracic cavity. So we mentioned that the muscles in the rib cage and the diaphragm This allows the change of volume of the thoracic cavity. So, as the thoracic cavity and lungs move together, this changes the volume of the lungs, and in turn, that changes the pressure inside the lungs. So, as I mentioned, Boyle's law states that when the volume of a gas is inversely proportional to the pressure, and in this case, temperature is constant. So you have PV equal to K, right? P and or P1 V1 equal to P2 V2. You guys would have studied this, right? So when the volume of the thoracic cavity increases, so you have taken in a breath, the volume of the lungs increases and the pressure within the lungs decreases. And when the volume of the thoracic cavity decreases, so essentially your diaphragm is back to being dome shaped, and the rib cage is contracting, the volumes of the lungs decreases and the pressure within the lung increases, so that forces the air out of the body. Okay, so that's how this law in physics actually regulates how air is going in and out of our lungs, right? So inspiration essentially is the phase of ventilation in which air is entering the lungs, and you have the diaphragm here, okay, that flattens out, extending the dimensions of this thoracic cavity. So it moves, it flattens out, so it presses against the abdomen, whereas this region is allowed to expand. And then you have these intercostal muscles. So the external intercostal muscles, they elevate the ribs and the sternum. So this is the sternum. This is the bone at the front of your ribs, right? You can feel it even with your hand. And you have these ribs that form the rib cage. And these muscles are sort of in between them. And what these external intercostal muscles do is that they contract. And when they do that, they sort of pull um, the ribs and the sternum to allow elevating them. So elevating means that they just move upwards, and that extends the anterior posterior dimension, meaning this is anterior, this is posterior, right? So this dimension increases. So this uh, distance here would increase because these move outwards. The ribs and the sternum move outwards. Okay. And so the action of these inspiratory muscles results in the increase in volume of the thoracic cavity. And the lungs, as we mentioned, there's this pleural seal, right? Because of the fluid there. They're held against the inner thoracic wall by the pleural seal. They also, therefore, undergo an increase in volume. So, along with this, the lungs also move, and the membranes are pulled outwards and create an increase in volume of the lungs. And then, of course, Boyle's law is followed, and the pressure gradient is followed. And If when during inspiration, the pressure of the environmental, like the external environment with respect to lungs is greater, and then in during expiration, it's the other way around, right? So the lungs are, or the thoracic cavity is contracting, diaphragm is dome shaped, and the gradient is towards air moving out of the body. Okay, so I hope that is clear to everyone. So yeah, so that's like the sort of gross like mechanics of breathing, and now we'll talk a little bit about these different types of volumes, which are very important in measuring whether someone has healthy breathing or not. So these different types of volumes are measured by physicians using spirometry to determine whether someone's breathing and their flow of breathing is normal, or is there something wrong at some stage. So this is a trace from a spirometer, and this is from this yeah, this is a really nice site, so you guys can check it out. And this, yeah, so this is this trace from a spirometer that shows the change in the lung volume. As so, imagine there's a patient and they're breathing normally, and they are asked to perform a few different things. So the physician will tell them, okay, breathe deeply, okay, exhale like this, okay, hold your breath, whatever, right? So, and then while they do that, these readings will essentially show, and they're like a readout of lung volume in milliliters. Okay, so the first part here that you can see, these small oscillation waves, these are this is known as tidal breathing. So it's the resting. So the resting state or the tidal breathing, that's just when a person is breathing normally without, trying to exhale excess volume or take in excess volume of air or and do any kind of special antics. That time, this volume inspired during each normal breath is referred to as tidal volume. Okay, so this is the tidal volume part. Now, uh, when normal expiration, so normal inspiration and expiration is taking place, when normal expiration is taking place, the lung is actually not empty. So it makes sense, right? We're not actively exhaling all the air in our lungs constantly. There is some amount of air left. So, um, and we'll talk about that. So when this patient, for instance, breathes out as far as they can. So normally during tidal breathing, you're breathing out some normal level. So now if you guys just sort of think about how you're breathing, right? You're just breathing in normally. You're not putting in an excess effort to remove all the air as much as possible. But if you did that, suppose now this patient is doing that where they are trying to throw out as much of air as possible from their lungs physically, this and that excess air that comes out over the tidal amount, that is called expiratory reserve volume. So just from the name you can tell that in tidal breathing you will have some x amount of volume left, but if you put an effort to throw out as much air excessively over tidal volumes or tidal amounts, then that extra air that you're able to remove is called expiratory reserve volume. So you can see that over here, right? And then, even at this point, even after removing expiratory reserve volume, your lungs are still not completely empty. There is some amount of air still remaining in the lungs, in that patient's lungs, in your lungs. And you can do this right now. You can try this as well, right? You're breathing normally, and then you take, like, after a normal breath, if you're like breathing out with as much force as possible, like that, right? Then you're actually removing your expiratory reserve volume, and even then there's some air left. That is your residual volume. So that's over here. Okay. Right. Then, okay. So basically, you can never completely empty your lungs. They're never fully empty. There's always going to be some air left, and that air is obviously not pure oxygen or pure carbon dioxide. It's a mixture of gases. Okay. So that residual volume is going to be there no matter what you do. Now, if the patient takes a very big breath, so imagine you take a much bigger breath than normal, so much bigger than your tidal volume, you take some really big breath. This extra volume over your normal tidal volume of inspiration is called inspiratory reserve volume. Makes sense? Over your tidal breathing, if you expire, like if you breathe out extra, that's expiratory reserve volume. If you breathe in and take a really big breath, that is the extra over the tidal volume is your inspiratory reserve volume. Now, relating to this volume is inspiratory capacity. Okay, so this is the volume that you can take in to the lungs after a normal expiration. That is over here, inspiratory capacity. Okay, so that's sort of related. And then another valuable measurement, apparently, to the clinic is the vital capacity. So, this you would have heard of before. This is basically the volume of air that a patient can move out of the lung after a full inspiration. That is the total lung capacity. So, you do a full big breath and then you move the air out. So, obviously, you always have to minus the residual volume because that's not going anywhere. So, essentially, it is the volume, the total volume of air that can move out of the lung after one huge inspiration. That is the total lung capacity. So these are just measurable quantities. Like we've already discussed the main things. Now we're just sort of looking at what is easy to measure. So tidal volume, expiratory reserve volume, residual volume, okay, inspiratory reserve volume, and then you have your total lung capacity, and then you have something called the forced vital capacity. So this is a common measure apparently when they're testing for if the lungs are healthy, and this is simply the volume that can be expelled from total lung capacity during a forceful expiration. So you forcefully move your air out, like you breathe out as hard as you can, and that is the total volume that you can throw out during that time. That is your forced vital capacity. Yeah. So in just normal vital capacity, this is just after a full inspiration, how much you can breathe out, while this is how much you can breathe out after a normal inspiration, but with force. So it's like you're trying to also get rid of your expiratory reserve volume. So these are more sort of metrics that are used to that measure and tell about lung health at the level of the clinic, and these are physiological terms for more exact um, measurements. Cool. So that is about these different types of volumes, and I'm sure we'll discuss them when we go through some questions as well. So let's move forward into some questions. Does anyone have anything that they're confused about? Anything that they want me to repeat? Anything? Let me know. Oh, okay. Okay, I think it's fine. Yeah. All right. Huh. Okay, so let's start with some questions, and yeah, we can discuss as we go. So when breathing stops with an open glottis, what happens to the alveolar pressure? So what do you guys think? Right. So I think you guys have tried. Okay. Okay. Right. So the right answer here is absolutely right. It's option C, which is equal to atmospheric pressure. So what's going on here is that when airflow stops, the alveolar pressure becomes equal to the atmospheric pressure because there's no pressure gradient anymore. There's no airflow, right? So in that case, everything will equilibrate and um, but the glottis is open, meaning that the air is sort of equilibrating in the entire respiratory canal. In that case, the alveolar pressure will become the same as the atmospheric pressure. So I think I didn't mention what the glottis actually is before. I realized like I should have mentioned that when we were talking about the anatomy of the respiratory system. So the glottis essentially this opening that's there in the vocal folds. So it's inside the larynx. So it's not the complete larynx, it's just a little space between the vocal folds. So essentially, the air pathways like the air comes through your nose and your mouth, it goes to the pharynx and then the larynx, and then through the glottis within that, and then it goes to the trachea and the bronchi, and then so on and so forth. So that's essentially at the entry of the trachea between the larynx and the trachea is where the glottis is. So it has some vocal folds and this little space in between them. So during like quiet breathing. So essentially, when you're just breathing normally, this glottis is open and air is nicely moving into the trachea. And this, like you being able to like make proper sounds when speaking and so on, like the glottis is very important for that as well. So it regulates like the amount of air passing through and like the vibrations that are produced to produce sounds. Um, and also, oh yeah, during like to make sure that food doesn't go into the windpipe. So the epiglottis is important because it like folds down and closes. And the glottis also closes while you're eating, so it prevents food from entering the airway. So it prevents what's called aspiration, which is like food going into the airway, which is really dangerous, right? And the glottis closing can like make sure to maintain like pressure. So if um, so essentially, if the glottis closes, it can increase intrathoracic pressure. Does that make sense? Because if you're closing it, you're essentially creating like a closed chamber in the thorax. So if the glottis is closing, it can increase intrathoracic pressure, and then there will be equilibration within that. But I don't think it closes like for a long time. So during breathing, like normal breathing, it's open. If you're, deeply, taking in breaths and breathing hard, it's still open. When you're speaking, it's like it'll be like close to some degree because the air that is reaching into your larynx needs to be regulated so that sounds can be produced properly. And while you're swallowing food, when you're eating, it is closed. So that's why if you're like eating and talking at the same time, you can like aspirate some food sometimes and start coughing because there'll be like a malfunction there where your glottis, epiglottis, they're not regulated well enough that you start choking on your food. So yeah, cool. uh right. So in this case the answer was option C, which is if breathing stops and the glottis is still open, then alveolar pressure will equilibrate with atmospheric pressure because there's nothing stopping the air within the cavity. Okay, now if you have normal intrapleural pressure during quiet breathing, what is it approximately? So this is just sort of a fact question. If you guys know, do send your answers. Right. So the answer here is actually B option, which is -5 to -7 cm H2O. So that's the pleural pressure. So why is it negative, right? So it's negative because, like, if you have, like, if you look at the lungs, when they recoil inwards. The chest wall will pull them outwards. So this creates like a suction, right? That keeps the lungs open. So suction is pressure, or like, kind of vacuum pressures are typically annotated as negative, right? So it's a negative pressure. So that's why. And the values are about -5 to -7. So that's just it. It's just the way it is. Okay. So the right answer here is B option. Um, yeah. Okay, I think I gave away the next one. So the primary role of negative intrapleural pressure is which of these? So I think that is now kind of um, kind of obvious. So give it a shot if you guys know. Yeah, exactly. So let's not spend too much time on this one because it's very clear, right? So the answer is option C. It keeps the lungs expanded against collapse, right? So yeah. So negative pleural pressure would, make sure that the lung expansion is maintained and the lungs don't collapse because of the pressure. So if you if there's a loss in this negative intrapleural pressure, so if there's some problem, maybe if someone has got into an accident or something or some medical condition, that loss can lead to a condition called pneumothorax. Um, so I think if you guys have watched any like medical shows, they love to use the word pneumothorax all the time with patients. So yeah, that's what happens over there. Yeah. Okay, great. So let's move to the next one. So 12 was option C, in case anyone missed it. Let's go to the next question. So during normal inspiration, alveolar pressure typically changes from what value to what value? So remember what we talked about with respect to partial pressures and gradients. And think about which direction the air needs to flow, and that will hopefully reveal the answer to you. So, okay, so let me explain this one. So during normal inspiration, so inspiration means we're taking the air in. So the air needs to move uh, um, from like the atmosphere into the alveoli into the blood. So for this, you would need a pressure gradient between um, like the respiratory tract and the alveoli and the blood. So you would have essentially you'd need like a suction, right? You'd need a negative pressure that allows the gas to move into the alveoli during normal inspiration. So actually the alveolar pressure falls a little bit, but it needs to be a suction pressure because it needs to pull the air in to the alveoli, right? So you essentially have um, a very, very small pressure difference. Just a small pressure difference is enough. So like going from 0 to -1 is enough. Um, so in this case, the answer is B, which is 0 to -1. Just that amount of change is enough alveolar pressure difference or pressure gradient, and that allows air to move efficiently. So the option here is B, and it's negative because it's a suction pressure. Okay. Okay. Okay, so this is about the volumes that we discussed earlier. So the volume of air inspired or expired during normal or quiet breathing is called which of these? So I explained it. Do you guys remember? Okay. Does anyone else want to give it a shot? Hmm. So the correct answer here is option D, which is tidal volume. Okay. So I'll explain this, and I can understand why it might be confusing between tidal volume and vital capacity. So tidal volume is the air that sort of moves, is inhaled or exhaled during normal quiet breathing. Okay, so uh, normally, it's some around half a liter per breath, and this is at rest. So it's automatically this is what's happening. So this is your, you don't have to put any extra effort for it. So everyday breathing amount is the tidal volume, whereas vital capacity is the maximum amount of air a person can exhale after taking in the deepest possible breath. So imagine you take in like a huge breath, like take in as much air as possible, like a really deep breath, and then you exhale. So essentially, it is the, if you add up the tidal volume and then the inspiratory reserve volume and expiratory reserve volume, that's when you get vital capacity. So depending on, a person, their health, their gender, age, fitness, whatever, like or even their like size, physical size, it can be like 4 or 5 liters. And this is important measure in spirometry. So you have to do forceful breathing with a lot of effort for measurement of vital capacity, right? So yeah, that's cool. So that's the difference between tidal volume and vital capacity. Okay. So if you were to for instance think of, like, imagine that the lungs are like a balloon, then the tidal volume is like you just blow a little bit of air and then the air comes out and that's it. Okay. But in vital capacity, it's like you fully blow air into the balloon and then you deflate it completely or as much as possible. That's the vital capacity. Okay. So if, for instance, someone's sick, they have, pulmonary fibrosis, right? Where you have, essentially it's a restrictive disease of the lung or some sort of muscular problem, then vital capacity would be reduced because they're not able to inflate, and sort of this elasticity of the lung is greatly reduced. Cool. So anyway, this answer here, the correct answer is tidal volume, which is D option. So I hope that's clear, and now you know the difference between vital capacity and tidal volume. Okay, they're both very important for making sure that someone has healthy lungs, but they are different metrics. Okay, now, all right. So question about transpulmonary pressure. What is it referring to, and what's the difference that it refers to? Hmm. So, um, yeah. Does anyone want to try? I can go ahead and tell the answer then. Yeah, yeah, good job. That's exactly right. So the answer is B. It is the difference between the alveolar pressure and the intrapleural pressure. So essentially, it is what determines lung expansion. So your PTP or transpulmonary pressure is equal to your alveolar or P alveolar minus the P pleural. So that determines the lung expansion that takes place. So that's what transpulmonary pressure is. I think I have some more questions. Okay, so this is about lung compliance. Important, very important parameter. So what is it defined as? We'll talk about it in some detail now. Does anyone want to try? Anyone remember what lung compliance is? Yeah, yeah, great job. So you're absolutely right. It is option C, which is the ease of expansion. So, like the ease with which lungs expand in response to a given pressure change. So if there is high compliance, if it's easier to inflate. If there's low compliance, the lungs are stiffer. So um, how much essentially volume change does the lung have for a given pressure change? So practically, if say lungs, okay, have I put a question for this? Okay, I have put questions for this, so I'll go into detail after the next one maybe. So what do you think reduced lung compliance is commonly associated with? So we'll discuss about high and low compliance after this. Anyone? Okay. Yeah, so you're absolutely right. The answer is indeed option B, which is um, reduced compliance is commonly associated with stiff lung tissue, which can be caused by fibrosis of the lungs. So what does this actually like mean, right? So if the lung, so as I mentioned, compliance is essentially the ease with which lungs expand when there's a pressure change. So if you have low compliance, it means that even if there's like even a large pressure change, there's only a very small volume increase in the lungs. And that's because the lungs are stiff and they're harder to inflate. So pulmonary fibrosis is one such condition in which case the lungs have low compliance. So they're just, you can imagine that they're just not as stretchy. So it's like the balloon, like if you go back to that balloon analogy, that balloon, even if you're putting a lot of effort, you're blowing air into it, there's something wrong with the balloon that's not allowing it to completely inflate. Right? So it's like a thick rubber balloon, maybe, that if you're blowing air into it and it's just not expanding, that's because it has reduced lung compliance. And high compliance, on the other hand, is when even a small pressure change can cause a very large volume increase. And in fact, they might remain increased. Okay, so they expand easily, but they don't recoil well. So that actually can occur in emphysema. So it's like people who have emphysema complain that it feels like their lungs, they're just there's just air inside and it's not coming out. It's actually because of that. It's because their alveoli are not like properly stretchy anymore, so they become inflated and then they remain inflated, and so that gaseous exchange, that gradient is not set up properly, and that's because of high compliance. It's because they expand, but then they don't recoil well. That may be the case if there's someone who's sick. Um, so in this case, a small pressure change can actually trigger a very large volume increase, and that then the recoil back to basal state may not occur as it should. So that's about lung compliance and the condition where it's high or low. Um, Yeah. Okay. Apparently, I have another question about the different types of volumes that you can measure in the lungs. So, after maximal expiration, right, the volume of air that remains in the lungs, regardless, is called which of these? So, we talked about this. So, let me see if you guys remember. Yes, good. Does anyone else want to give it a shot? I'm happy to see that you guys are all getting it right. So, it is the residual volume. That is the air that remains even after you've put in full effort for maximal expiration. there is just a tiny bit of air left, and that is called the residual volume. So that's option D. So this residual volume, why does it need to be there, right? Why can't we just have zero air left in the lungs? Because this actually prevents lung collapse and it prevents like fluctuations. You have to remember that the lungs themselves are wet on the inside. So if you don't have any gas left in the lungs, what's going to happen, right? Remember about surface tension. So you remember that the layer of air inside, this was, sorry, layer of water inside the alveoli is what causes surface tension, which can cause them to collapse. In the lungs also you have air, and that prevents the membranes from touching each other because the membranes are wet and they have water, which would cause surface tension and inward force that would cause the membranes to like stick to each other, right? But having a little bit of air remaining prevents lung collapse. So that residual volume is actually life saving. It's got to be there. And that residual volume is not only in the lungs, it's also there in all of the, like the bronchi and bronchioles and everything. It prevents their collapse as well. It's not only is it the capillaries, the cartilage that prevents it, but also there is some amount of air that remains in them as well. Cool. Okay. So that's about residual volume. Okay, apparently I have some more questions and then I have a few more theory points that we can discuss. Okay, so the approximate resting lung volume around which tidal breathing occurs, I mentioned this in brief when we were discussing tidal breathing. Do you guys remember what volume is the resting lung volume during quiet or tidal breathing? Okay, I think I have some answers here. Hmm. Okay, so the right answer here actually, and I can see why one might confuse it with what is the tidal volume, but those two are actually two different things. So the answer here that I got is 500 ml. That is the tidal volume. Okay, but resting lung volume is actually something a little different. So 500 ml is the tidal volume. You guys got that right. But what is resting volume, lung volume? It's also called functional residual capacity or FRC. Okay, it is the amount of air present in the lungs at the end of a normal quiet expiration. Okay, so when you haven't put in any extra effort to breathe outwards, then whatever air is left, that is the resting lung volume or the functional residual capacity. And the answer to that is actually 2300 ml. So option C, 2300. So let me explain how this is working. So when there is air remaining in the lungs after normal tidal expiration, you're not forcing any air out, you're not inhaling very deeply. It's just a rest between two breaths. So what does it include? It includes the expiratory reserve volume, meaning the amount of extra air that you could have breathed out, plus the residual volume, which is the air that will always be there. Right? So if you add these two quantities, that is your resting lung volume. Okay, so tidal volume is different. That's just the amount of air that you're breathing in a normal tidal breath. But this is how much is left in your lungs after you have removed one normal breath. So in a normal adult, typically it is around 2.3 liters. Okay, and this is important because it's like a mechanical equilibrium point. And yeah, so now if someone has some disease, of course, then this FRC or this resting lung volume is going to change. So, like in emphysema, because it just feels like there's air inside, and it's the elastic recoil is lost um, in the alveoli. The FRC increases, fibrosis. The lung is not expanding properly, so it's decreased. If someone is lying down versus someone is standing, your resting lung volume will be different. If you're lying down, it's actually decreased. So the amount of air that's remaining in your lungs is decreased. But if you're standing, it increases, relatively speaking. So, yeah, here the answer is 2300. Okay, so your tidal volume is 500 ml, which is essentially how much you're inhaling in a normal quiet breath. That's 500 ml of air, and you exhale also around 500 ml, but the lungs will continue to have 2300 ml of air after that. Okay, so I hope that is clear to everybody. So that's the difference. Okay, so I think you guys had answered mostly 500 ml. So now that difference I'm hoping is clear between what tidal volume is and resting lung volume is. Okay, so the answer here was option C. Okay, air within the lungs is not completely replaced with every breath. And what is the reason for this? This is, I guess, one of several reasons. So what is one of several reasons for this? So I think I'll go ahead with the answer so that we don't run out of time later for the other things. So the right answer here is option B. So which is to maintain stable temperature and humidity in the alveoli. So when you have air mixing that's happening when you're breathing, it's gradually it maintains constant temperature. So for instance, if you were to stand in a warm humid room and then suddenly walk into a very cold and dry room, and you keep breathing, immediately all the air in your lungs is not going to become cold and dry, right? You're still going to have some of the warm humid air in there, and there's going to be gradual mixing as you start breathing in the different air. So that allows some amount, so it's not like this sharp change that shocks your system. You have a gradual mixing that helps to equilibrate and try to maintain, because it's not like your body would not adapt to that either, right? You would have some changes in your body that would try to maintain equilibrium with respect to temperature, humidity, yeah, and so that would help like protect alveolar cells, which they're really delicate, these alveoli, right? They don't have like much support because they don't have like cartilage or anything. They're just really thin membranes. So you want to make sure that they're not undergoing a lot of stress. Um, so having some air remaining that's already equilibrated and then mixing slowly with the new air allows to maintain stable temperature and humidity and other conditions in the alveoli. Okay. Okay. All right. Last question. So gas exchange within the alveoli mainly occurs by um, which mechanism? Right. So you're completely right. The answer is indeed option D. So that's diffusion without turbulence. So there's no, at the level of the alveoli, obviously there is no turbulent airflow, and gas exchange is only occurring by diffusion through the very thin membrane of the type 1 cells, right? So flow of air is actually only occurring in conducting airways. So in your bronchi and your bronchioles, you have air that flows. And there's actually, you guys, if you're interested in it, I do remember reading about it, though I don't know in too much detail. There's a group in NCBS that works on it, as well as I'm sure others, and they look at how like airway epithelia, like morphological characteristics, as well as their functional readouts, those change with respect to different regions in the respiratory epithelium. Um, and it's actually fascinating where I think I don't think they look at it, but like there's a mechanical aspect to it as well, right? Like you have air that's actually flowing against these cells, and like upper respiratory tract versus lower respiratory tract, the turbulence of the flow would be very different, and that mechanical effect of air could actually have a physical like effect on the cells. So the cells actually adapt to the flow of air, and evolutionarily also I think that there have been adaptations that allow people living in different environments and conditions to adapt to these kind of difference in air quality and pressure around them, and that could actually change the respiratory epithelia like at the cellular and morphological level. So it's just interesting. I thought I'd mention that. Okay, so now there's, I think, one or two very important concepts that we should cover, which would help you out in your assignments and in general understanding, furthering your understanding here. Which is one is this VQ ratio, and the other is we'll just talk briefly about hemoglobin, right? So yeah, so let's, what is this VQ business, right? So to ensure adequate matching of ventilation and perfusion of the lungs, there has to be and when this matching takes place, um, this will allow the continuous delivery of oxygen and removal of carbon dioxide from the body, right? So that's the goal of of breathing, right? That you are continuously taking in air that then delivers oxygen to your tissues, and then the carbon dioxide produced at different cells and tissues is then carried back and thrown out from the lungs. And there is a physical sort of matching of these two parameters of ventilation and perfusion at the lungs that allows this delivery mechanism to take place. So what is this ventilation or ventilation rate? So we'll call that V, okay? This refers to the volume of gas inhaled and exhaled from the lungs in, say, a given time period. So usually one would calculate over a minute. So this is calculated. So you multiply the tidal volume. So now it's good that we talked about what the tidal volume is, because it is about 500 ml, right? So the volume of air inhaled or exhaled in a single resting or quiet breath, and you multiply this by the respiratory rate. So in an average man, of course, this would be different for maybe, even between men, and obviously between men and women. The ventilation rate could be calculated roughly as 6 liters per minute. So you multiply 500 ml with the rate of respiration within a minute. So like how many times they have breathed in and out. Then you eventually land up with 6 liters per minute. So every minute there's 6 liters of air that has been fluxed. So that's your ventilation rate, or V. Okay. Then you have this component called Q, which is your perfusion. So perfusion is, with respect to the lungs, this refers to the total volume of blood reaching the pulmonary capillaries in a given time period. So pulmonary capillaries are the capillaries that are surrounding your alveoli, and they are the ones very important for the actual gaseous exchange. Okay, so the total volume of blood that reaches these capillaries in any given time period is your perfusion, or your Q. And so now if you have ideally, if you look at this ratio of V to Q, or the VQ ratio, ideally 1 is best for maximally efficient pulmonary function, meaning that um, the amount of air that's been inhaled or exhaled uh, for within a minute is equal to the amount of blood that has reached the pulmonary capillaries, essentially meaning that continuous delivery is taking place. But this ratio would also vary not just in different situations, like if someone has some disease or if they're, doing some exercise, but it could also vary depending on which part of the lung you're looking at. So, for instance, when a person is standing up straight, the ratio is different in different parts of the lung. So, at the apex of the lung or the top part, it's about 3.3, meaning the ventilation rate is 3.3 times the perfusion rate or the perfusion. Whereas towards the middle, it's more closer to 1, right? And at the base, it's the lowest. So meaning that perfusion is higher, but respiratory rate or ventilation rate is lower. Perfusion is higher because of gravity. So we'll talk about why. So in this means that in the apex you have ventilation exceeding perfusion. So V is more than Q towards the apex, and towards the base you have higher perfusion than ventilation, and it's because of the gravitational force that pulls fluids downwards. So even in the lungs you have this gradient of VQ ratio. Okay, and that's also sort of um, demonstrated here, which is if you move with respect to the ribs. So this is the top and this is the bottom. So this is the second rib, fifth rib, whatever. And then you see that your VQ ratio, your perfusion, when you move from top to bottom, your perfusion is increasing because of gravity. So your Q is increasing, whereas ventilation is not increasing as steeply. So that's why your VQ ratio is also accordingly, it is different. It's at the top, it's closer to 3, and at the bottom, it's less than 1. Yeah. So to explain this a little more, so there's different ratios for different areas is because of the relation of the area to the heart as well as because of gravity. Okay. So the areas of the lung below the heart have increased perfusion, right? So you saw it here. Area of the lung below the heart have increased perfusion, meaning a lower VQ ratio. And this is because of gravity, the fluids are going to be higher in the lower regions. So this reduces the VQ ratio. And actually, in the human lung, overall, if you look at the sort of general overall value or average value, it's actually closer to zero point 0.8. So ideally, one would want 1, but your whole lung, if you look at it and look at the gradients of VQ ratio between the top to the bottom of the lung, the average is around zero point 0.8. And gravity triggers these changes in ventilation and perfusion because pleural pressure is increased at the base of the lungs. Why? Because there's more liquid pooling over there and because it's lower than the heart. So, because of gravity, the pleural pressure increases. So, resulting in more compliant alveoli and increasing ventilation. Whereas hydrostatic pressure is decreased at the apex of the lung, resulting in decreased flow and decreased perfusion. So as perfusion increases with gravity, the apical and middle zones of the lung that we saw here, apical and middle zones, they see the greatest relative increase in their perfusion rate with an increased cardiac output, like for instance during exercise. Okay, so this is just showing that. In fact, I'll explain it in the next slide. So now if um, alveolar ventilation. So imagine alveolar ventilation and alveolar blood flow are not matched with each other. This would actually be reflected in the VQ ratio that's measured. So if there is inadequate ventilation, right, that means there is inadequate, like when you're breathing, the amount of air that you're breathing, that 6 liter per minute for the adult that we saw, that is actually decreased for whatever reason, right? Then the VQ ratio would reduce. So the gas exchange within the affected alveoli is impaired. So as a result of that, your capillary partial pressure of oxygen or your PO2 would also fall because enough gas is not getting in. and the partial pressure of PCO2 would rise because enough carbon dioxide is not getting out. Now, in response to this, if this is the case, then how would the body try to compensate, right? So you have something called hypoxic vasoconstriction that would cause diversion of blood to the better ventilated parts of the lungs, because again this gradient is there in the lungs. So in most physiological states, hemoglobin in these well ventilated capillaries will already be saturated, so there's only so much the body can do, right? Because you cannot suddenly increase the amount of red blood cells and hemoglobin in your body to compensate for this kind of fall in PO2 and increase in PCO2. So actually the red blood cells will only be able to do so much to improve binding through additional oxygen to increase PO2. So after a point, then there'll be saturation and the PO2 of the level will continue to remain low, even though there might be some initial relief because of movement of blood to other regions of the lung. And that would act as a stimulus to cause hyperventilation, resulting in either normal, or if it doesn't work out at all, then there need to be some kind of medical intervention. So hyperventilation just means like breathing really hard. And a mismatch in ventilation and perfusion can arise due to reduced ventilation of part of the lung or reduced perfusion. So either you have some mess up in the V or some mess up in the Q. So here you can see something called shunt perfusion, which is there is perfusion without proper ventilation. So that would result in what? It would result in a low VQ ratio. So you have, this is your capillary and this is your alveolus, and here you don't have proper alveolus. So example is if there is like an airway obstruction, if someone has pneumonia and a lot of fluid buildup, so air is not reaching the alveolus properly. So you have like perfusion is fine, the blood is flowing fine in the capillaries near the alveolus, but air is not coming in. That means V is low and Q is normal, so your VQ ratio is low. Normal condition, of course, we talked about how it's approximately zero 0.8. So you both have air here and you have proper um, blood in the capillaries here. So you have proper exchange. So you have O2 coming in and CO2 going out at the rates that are required. And finally, you have the case of high VQ, meaning dead space ventilation. So you have ventilation without perfusion, meaning you have proper air in the alveoli, but because of some reason or the other, you don't have blood flowing properly in the capillary. So it can be due to a pulmonary embolism, cardiogenic shock. Essentially, that just means that there is maybe a clot or some kind of obstruction that's preventing blood from moving into the capillaries near the alveoli. So in this case, perfusion is low and ventilation is normal or high, in which case your V by Q ratio is high. Okay. So anything that is deviating too much from your normal zero 0.8 is going to be some sort of, condition which needs some sort of intervention, most likely. Okay. And the next concept, oh, it's already 8:50. Huh. Okay. All right, I'll just explain this. And the last couple of questions maybe we can discuss in the next class then, considering that time will run out. Oxygen is transported in the blood in two different ways, right? So it's dissolved in the blood. That's like the minority, and the majority of it, like 98.5% of it, is bound to hemoglobin, right? You guys all know about hemoglobin, right? So once oxygen has entered the blood from the lungs, it can be bound to hemoglobin that is present in the red blood cells, RBCs. So this is hemoglobin. Okay, look at its structure. It has this quaternary folded structure, right? You guys must have studied about it in biochemistry. And it's a protein that has four subunits. So it has two alpha subunits and two beta subunits. And each subunit has this heme group in the center that contains iron, and it binds one oxygen molecule. Okay, so that's the carrier, right? That's what's going to carry your oxygen. So this means that each hemoglobin molecule can bind four oxygen molecules, and that's when it's called oxyhemoglobin. And hemoglobin molecules with a greater number of oxygen molecules bound. So it doesn't mean that all hemoglobin at all times have all four oxygens bound. If they have more oxygen bound, they're brighter red, which is why oxygenated arterial blood, right, which has freshly been oxygenated at the lungs, is brighter red, whereas deoxygenated venous blood is darker red. Okay, because fewer oxygen molecules are bound to the hemoglobin. And when you have oxygen binding to hemoglobin, you can actually see the sigmoidal curve, which I'll talk about. So hemoglobin binding, when hemoglobin is bound by oxygen, there's a change of its shape, okay, based on how many oxygen molecules are bound to it. So the change in shape can actually change the affinity it has for oxygen. So it's like, yeah, so I'll give you an analogy after this. So the number of oxygen molecules, as it increases, the number of oxygen molecules bound to hemoglobin, as this number increases, the affinity for hemoglobin for oxygen increases along with that. So this phenomenon is called cooperativity. So it's like, for instance, if you, it's kind of a silly analogy, but suppose you're really hungry and you eat like one piece of food. So you eat one French fry, and then you're like, oh my God, I want more. And so your need for French fries increases a lot. And so you eat another one, and then you eat another one. And obviously, at some point, it's going to saturate. But until then, your affinity for French fries as you eat them is going to increase a lot. So that's like a cooperativity analogy. So when no oxygen is bound, the hemoglobin is said to be in the T state, which is the tense state, and it has obviously low affinity for oxygen. And when your first oxygen binds, the shape begins to alter and it goes into a relaxed state, or R state, which has a higher affinity relatively for oxygen. And so if you plot this change on a graph where you look at oxygen saturation over the partial pressure of oxygen, eventually it starts increasing and eventually it flattens out because that's where it saturates. So the percentage of oxygen bound to hemoglobin is related to the partial pressure of oxygen, which is your PO2 at a given site. So when oxyhemoglobin reaches a tissue with a low PO2, for instance, like in your skeletal muscles, it will dissociate into oxygen and hemoglobin, and this releases the oxygen, okay? And that leads to a local increase in PO2. And when it reaches tissues, say, that has a high PO2, which is like in the pulmonary circuit, which already has a lot of oxygen, then hemoglobin will continue to take up more oxygen until it can't take up any more, resulting in a relatively lower PO2. So it takes up. So this is how hemoglobin is regulated in different tissues based on the local partial pressure and how it takes up the optimal amount of oxygen required to then release it into tissues that have relatively lower PO2. Okay. And there are some factors that can affect this affinity. So the pH. So I think, uh, yeah, I'll discuss this again in the next class and we can go over it in more detail. But the pH and certain compounds, temperature, and so on can increase or decrease this. And there's this effect called the Bohr effect, which we'll go into, I think, in the next class. And then we'll discuss these question answers as well. We'll talk about carbon monoxide poisoning. Yeah, so I think for today I will wrap it up because it's getting late. Um, oh, sorry. Okay. So if anyone has any other questions, please let me know. Um, and we'll continue this with in the next class, right? All right. All right. Yeah, great. Thanks all for joining. Good night, and see you in the next one.